And the final topic for this course is going to be nucleic acids. These are something that you commonly know as DNA and RNA. And in this course, we're going to keep it fairly simple. We're going to focus on some of the chemistry and some of the physical properties and the structure of these nucleic acids. Chemically, nucleic acids are polymers of nucleotides. Therefore, the nucleotides are the monomer that creates the nucleic acids, RNA and DNA. Those monomer nucleotides are actually fairly large molecules themselves. They are composed of three major components. They are a sugar, a phosphate group, and a nitrogenous base. Let's look at each of those three parts individually. Let's start by looking at the sugar. So we're going to look at both RNA and DNA, and we're going to look at them side by side. The R in RNA stands for ribonucleic acid, and the ribo tells us that we're using ribose. The structure of ribose is a 5-membered sugar with an oxygen here at the top, and then all these oxygens coming off of it, and that is ribose. It's 5-membered sugar with OHs on every position, and again our oxygen in the top there. When we look at DNA, it is deoxyribose. And you'll notice in its name, it's saying that an oxygen has been removed. And that's going to be in position 2 of the ribose. We've taken away the oxygen there, and now it becomes deoxyribose. So ribose is found in RNA, and deoxyribose is found in DNA. That loss of an oxygen changes the physical properties that are found for DNA from what they were in RNA. Mostly what we're going to notice is that there is fewer OHs, which means less hydrogen bonding. That means that this is more hydrophobic than RNA, and so we're going to see its properties shift just because of the loss of that OH. But before we continue with the overall structure of RNA and DNA, let's finish up with the structure of the nucleotides that make it. Our next group is going to be the phosphate group. Now you've seen phosphate previously back in Unit 2. Remember the phosphate ion was PO4 with a 3 negative charge. And that's very similar to what we're going to see here. In this case, though, we're going to have it actually bonding to the sugar, so we're going to see that it has a bond there, so it doesn't have that negative charge where there's a bond to the sugar. And so it becomes PO4 negative charge, and that is going to be our phosphate ion. So at this point we have a sugar and we have a phosphate. What's important about the sugar and the phosphate is that they form the backbone of the nucleic acids. So when you have a nucleic acid, it's going to have a sugar-phosphate backbone. So it'll go sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And those are going to continue down, making the backbone of both the RNA and the DNA. Again, the only thing that's changing here is whether it has ribose or deoxyribose. Otherwise, it's just sugar, phosphate, and so just the sugar's changing. Now, what happens when we add on that last part, which is the nitrogenous base? What do we see there? Let's start with what is a nitrogenous base. A nitrogenous base is a basic substance that has a nitrogen in it. So this is going to be something similar to NH3, which we've seen before as ammonia. And in Unit 2, we said that ammonia was our only weak base. But as you know, in Unit 4, we learned a little bit more about organic chemistry, and we said that things with amines in them are weak bases. We're going to see the same thing with these nucleic acids and the nucleotides. They're going to have these things called nitrogenous bases. In the nucleic acids and in the nucleotides, we see there are only 2 subsets of those nitrogenous bases. The first type are called pyrimidines. The pyrimidines tend to have a 6-membered ring with several nitrogens on them. As you can see in each of these, there's 2 nitrogens in the ring and a C-double bond O coming off at least one other position. And in the nucleic acids, there are 3 common pyrimidines. They are cytosine, uracil, and thymine. And the second type of nitrogenous base are the purines. Now the purines are a little bit larger. They have the 6-membered ring that's similar to the pyrimidines, but then they have an attached 5-membered ring off to the side. The 2 common ones are adenine and guanine. So those are the three parts of the nucleotide. You need to have a sugar and a phosphate, and those form the backbone of the DNA or RNA. And then we'll also see the nitrogenous base. So three parts of a nucleotide: sugar, phosphate, nitrogenous base. Now we're going to focus on the similarities and differences between RNA and DNA. The first thing that we will see is in the sugar. In the RNA, they have ribose. In the DNA, it has deoxyribose. And again, that's a difference of an OH group. Next, we see that phosphate will be the same for both of them. And then we look at the nitrogenous bases. Both RNA and DNA have adenine, cytosine, and guanine. But where we see a difference, RNA has uracil and DNA has thymine. So that is a difference. We find uracil only in RNA, and we think thymine only in DNA. Next, we can look at the basic structures. We see that RNA has only a primary structure. Now, primary structure is the order of those nucleotides that they're held in. So here what we see is that the sugar and phosphate are joining together to make this long strand, and those nitrogenous bases or nucleobases are sticking out. And again, the primary structure is the order that those nucleobases are found in. While RNA only has primary structure, DNA has primary, secondary, tertiary, and sometimes people will even state that it has quaternary structure. So let's go through each of those. Secondary structure comes from those nucleobases that are attached to the long strands, joining together, and when they join together, the two strands become joined, and we get secondary structure, which is the fact that we have two strands. The tertiary structure is what you see here, where we see this coiling shape. That coiling shape is considered tertiary structure. You may have heard that DNA is a double helix. What they mean by double is the fact that there are two strands, and then the helix is this coiling shape, which looks like a spring. And finally, in quaternary structure, you might see something like this strand that likes to coil upon itself. As it coils up on itself, it shrinks in size so that you might see something like this. This is called supercoiling, and supercoiling is just as you tighten a coil, as you continue to tighten that coil, it starts to coil up on itself. And so that's what we're seeing down here is one of those supercoils, and that's very common with DNA, is it likes to supercoil around itself. When we compare the size of RNA and DNA, we see that RNA is much shorter. A typical strand of RNA has between 200 bases and 5,000 bases long. That's around 200 nanometers to about 30,000 nanometers long. On the other hand, we can see that DNA is a much longer chain, where they range between 100,000 base pairs up to about 250 million base pairs. In the human genome, it's going to be much larger, generally starting around 50 million base pairs. And when it's uncoiled, we see that DNA is somewhere between 2 and 8 centimeters long, with an average of 5 centimeters. When we look at where we find RNA and DNA, we find that RNA is found throughout the cell. It can be found in the nucleus and outside of the nucleus, whereas DNA is found only in the nucleus of the cell. It is never found outside of the nucleus. Now let's look at the comparative stability of RNA and DNA. Both RNA and DNA can be damaged by UV light, ionizing radiation, like X-rays, gamma rays, cosmic rays, radiation from nuclear reactors, that type of thing, and also from environmental toxins such as smoke, other carcinogens. When RNA is damaged, it's not such a big problem because it's just usually destroyed by the body immediately. And the body recycles the RNA over and over again. It's constantly being destroyed, so it's not a problem. RNA is easier to damage. It is less stable because it is single-stranded, so it's easier to be attacked. Everything damages it much easier, but again, it only hits one spot. It's not so bad. DNA, on the other hand, when it gets damaged, can cause much more severe problems. Because the base pairs are matched in DNA, it is very rare that there's a mutation in the DNA. In fact, it's about 1 in a million times that it happens. But even so, that amounts to about 10 new mutations every time a cell divides, because there are so many bases in DNA. And that means that in a typical day, it's estimated that 37 trillion cells in your body will accumulate trillions of new mutations every day. Luckily, our body has a system that allows it to fix those mutations, and it finds the errors and fixes them. Luckily, many of the codons that are in our DNA are useless, or as far as we know are useless, and we don't know exactly what they do, and so they have no effect if they get mutated. On the other hand, a small number of those mutations could strike a cancer gene, something that causes the individual cell to continue to divide and grow faster than it normally would. It ends up that one mutation isn't enough to make a cell cancerous, though. But it takes more than one mutation to allow this to happen. So first it has to mutate to cause the cell to overgrow. Then it also has to override the natural limit on how many times a cell can divide. The mutated cell also needs to escape apoptosis, the cell's tendency to self-destruct whenever something goes wrong. And it needs to evade an immune system that is constantly on the lookout for aberrant cells. Any single cell that has cancer must accumulate all these mutations to become successfully cancerous. But sadly, it definitely does happen in our world. And we can look at the different jobs that RNA and DNA have. We might say that the DNA is the brains of the operation, whereas the RNA is the brawn. So most of the work is actually done by the RNA, and we have several different types of. First, we have messenger RNA, whose job is to take the message from the boss, the DNA, and bring it into the ribosomal RNA, which is the factory where things are made. And so the messenger RNA translates the information from DNA code to RNA code and brings it into the factory where the proteins are actually made. And again, the major work of RNA is to make proteins in our bodies. Once it's in the ribosomal RNA, which we call rRNA, the ribosomal RNA is basically an area where reactions occur and which allows us to produce those proteins. Inside of there, transfer RNA will bring in various amino acids where it converts the information brought in by the messenger RNA and converts it into amino acids to form the proteins. And the proteins join together and they create your protein inside of the ribosomal RNA. And the protein grows out of the ribosomal RNA and starts to form and become active. So those are the major jobs of the RNA. The only real job that DNA has is to store information. So it's like a library. It holds the blueprint. It tells the directions of how to make every type of protein in your body. So DNA is vital, and it cannot be destroyed easily because if it makes a mistake throughout the whole system. If your RNA makes a mistake, when it makes a mistake, it makes it to one single protein. But if the library, the background, the blueprint, makes a mistake, if the DNA is damaged, that mistake will continue on creating damaged protein because it's the blueprint. Everything goes back to that, and the messenger RNA will always pick up the wrong message and create damaged protein. This image shows a simplification of what a cell might look like. So we have our cell in the center, enlarged greatly as our nucleus, and enlarged greatly inside of that is the double-stranded DNA coil that is found only in the nucleus. Also found in the nucleus is the messenger RNA. Notice that it is single-stranded. That messenger RNA will come in contact with the DNA and read the message that it has, transcribe it into the RNA language by making new base pairs that match in the RNA language where T is replaced by U. Next, the messenger RNA leaves the nucleus, goes out into the cell, and joins with a ribosomal RNA where the factory begins its work. And here we can zoom in on the ribosome and see that inside of it we have this chain of the mRNA, which is bringing the information from the DNA that's been transcribed from the DNA. It's now going to be read by the transfer RNA that we see up above. That's our blue one here, and we have two of them here. So in this one, I'm going to be taking two different amino acids. One has the reading from the messenger RNA is ACG. The transfer RNA is going to find its match, which is going to be, in this case, UGC. Attached to that will be an amino acid, and then inside of that ribosome we're going to have a second one. The next message on the messenger RNA is UGG, which means the transfer RNA is going to see ACC. Those two connect, and that means that those two amino acids are now held together well so that a bond can form, and that is the basic process. Now this continues down the chain for thousands of different amino acids until whatever protein it is made, but the basic concept stays the same. First those two join together, and then they're spit out the other end as the protein, and another one comes in and another one comes in, and the chain gets longer and longer.